So I do want to thank Westgrid for the opportunity to present today and to share a little bit about what I know about quality control of bioinformatic and next generation sequencing data. So my name is Kane Say. I work at the Candace Michael Smith Genome Sciences Center at the BC Cancer Agency. So just a little intro on what we do here at the Genome Sciences Center. We're actually a part of the Cancer Research Center of the BC Cancer Agency, and so here's a little slide that kind of outlines some of the accomplishments that we've had over the past, I guess, nearly 20 years. And actually, kind of looking at this slide, it looks like it's very similar to the way that I started my bioinformatic career around the turn of the century when I worked at the Saccharomyces Genome Database Project. So I guess back then, of course, you could sequence a whole genome, and it was a lot more significant. And with today's technologies, obviously, we're able to sequence genomes in a matter of days. So certainly, things have advanced a lot in the last 20 years. Taking a look at what kind of platforms that we use here at the GSC, we do have two main platforms, although the overwhelming bulk of what we do is on the Illumina sequence by synthesis instruments. We also have some Sanger capillary-based sequencing, and every month we typically construct around 15,000 high-throughput libraries with about 80 terabytes, greater than 80 terabases sequenced. On the bioinformatics side, we have 3 large-scale compute clusters, and you can see the specification there, as well as some team-specific clusters. So I'll talk about the BioQC team, which is the one that which is the group that I look over, and we have about a cluster with 220 cores and about 2.5 terabytes of RAM. And of course, we have a large amount of storage to store all of this data. So just to give you an overview of what I'm going to talk about today, basically what I'm going to hope to impart to everyone is that quality control is really a significant part of what we do here at the Genome Sciences Center. And what I hope that everybody can get out of this talk is to know the importance of quality control in terms of its relevance to you, as well as to the relevance not only to the work that you do within your organization, but also its impact upon the work that you publish and your science and all of the great stuff that we're all working on. I will also, in terms of the value, I'll also just sort of at the end, I'll cover a couple of some of the unusual cases of quality control that we encounter and what their impact might be. So what is quality control? So I like this quote that says, if you don't have the time to do it right, you're going to have to make the time to do it over again. And as I mentioned, at Genome Sciences Center, we spend a lot of time and effort, ensuring that the data that we produce is high quality. And we have, of course, all of our teams are responsible for monitoring quality. These do tend to be manual checks. And so what they do is they basically rely on the expertise of the operator, experience to sort of detect things and see if they can catch things before they come out or perform normal checks. But my team, the Bioinformatics Quality Control Group, we're actually dedicated to creating automated pipelines to monitor and report quality issues in an automated fashion. So why do we really care that much about quality? It's mainly because for 3 main reasons. One is to identify potential issues before the data analysis begins, to inform collaborators about their experiment, maybe tell them things that they may or may not have known about their system, and then finally, to improve our laboratory processes and our bioinformatics processes. So quality control is really defined as a system of maintaining standards in a manufactured products by testing a sample of the output against a specification. And so in the context of bioinformatics, manufactured products is basically the next generation sequencing data that we have that's coming off the sequencer. And the specification is basically what the type of experiment that the person was trying to carry out, whether it's a whole genome experiment, whole genome shotgun sequencing, or whether they're trying to do a capture experiment, or whether they're looking at microRNA or one of the many other types of experiments that we have. So it's important to make a distinction between quality assurance and quality control. So the team that I look after is the quality control, and we're really focused on detection of the issues, whereas quality assurance is more about prevention. And so I like this picture because it kind of shows the relationship between the two groups and that we sort of feed back to one another. So what we do is when we detect things, we investigate and we try to understand what happened. And then it's up to we report it. And then the quality assurance team will try to look at developing procedures or putting future safeguards in place or different things to ensure that it doesn't come up again. Quality assurance, we also have a quality assurance team here at the Genome Sciences Center, and that also, of course, plays a big role in all of our operations. But that won't be the focus of my talk today. I'll be more talking about on the quality control side of things. So there are different levels of quality control, and here they are framed in terms from a bioinformatic context. So level 0 quality control are non-alignment based metrics. In level 1, that's where we typically align it against a reference genome. And then in level 2 QC is an assessment after a some kind of bioinformatics analysis has been performed, whether that's variant calling or expression quantification or something else. So if we look at the levels of quality control and compare them to what the industry definition, and this is outside of biology or genomics, is that for level 0, level 0 quality control involves raw, unprocessed data that's directly observed on the instrument. And these tend to be absolute measurements. So if we look in terms of the bioinformatics, what that is input is basically the FASTQ files or the sequence files that we could get directly off the sequencing instrument. And this tends to be indifferent to protocol, so there's no metadata associated with this. So regardless of whatever pipeline was sequenced, we just get the sequence data, and that's our level 0 QC, and we look at that. The level 1 QC is what's considered to be quality control data. And so that's associated with metadata, and it's compared with calibrations, at least from an industry perspective. In the bioinformatics, we consider that to be aligned data, so we align that using a tool like BWA or NovoAlign, and compare that against standards. And to us, the standard would be, of course, our reference genome, whether it's a human genome or a mouse reference. And so there are different things like mapping rate and dupe rate and properly paired sequence. Level 2 QC, it involves derived products that require scientific or technical interpretation. And so these standards are defined by the community that collects or utilizes the data. And so, again, within the bioinformatics context, what that is assembly, expression levels, variant calling, on-target rate. Those kind of things are sort of what's called level 2 QC. So how do we perform quality control at the Genome Sciences Center? So we have many levels of QC, and as I mentioned, carried across different groups. For example, in the laboratory, we'll do DNA quantifications. We'll do Agilent traces to look at the sizes of the fragments. We'll do cluster density checks, intensity, focus scores, pass filter rate, PF rate, looking at the Q30 and Q20 scores, index splitting. For the level 0 QC, this is sort of the automated system that we currently use. We have over 60 metrics, and I've listed a couple of them here, but there's really, with 60, there's really too many to list on a single slide. And then at the level 1 QC, what we look at are things like alignment rate, and then for different experiments, like ChIP-seq experiments, we have specific metrics that are really only relevant to those, or bisulfite sequencing, or RNA sequencing, or microRNA. So they're different sort of metrics that are based on the alignment to the reference genome, but they're specifically meaningful to those experiment types. So let's take a look at the QC in the lab. Prior to sequencing, we have DNA quantification, and that basically is a determination of how much DNA is present within the sample. We also do a qPCR stage, which tells us how many fragments contain Illumina adapters. And of course, that's important because you want your Illumina adapters to allow the fragment to adhere to the flow cell so you actually get some usable sequence data out of it. On the instrument itself, the lab does a first base report, and they try to detect library issues or determine if these issues are specific to the instrument. And then we try to look for biased libraries, looking at the base calls, or reviewing the cluster density to see if there are any issues. And then immediately when the run is finished, they do a post-run QC where they look at the Q30 or Q20 scores. And this might give them information about the contamination of their cleavage mix or the temperature of the instrument if one of the temperature control units is not functioning properly. And they also look at index splitting to tell them things about uneven pooling or unknown indices. So once the data has been sort of QC'd by the lab, then it proceeds to level 0 QC. So why do we bother to perform level 0 QC? Well, we really have 3 reasons, one of which is that it's fast. So our goal is to QC all of the lanes that come off of all of the sequencers of the GSC within 24 hours of them being sequenced. It gives rapid feedback to the lab on a no-go or no-go decision for subsequent lanes. So if we have a project that's sequencing maybe 20 lanes, we'll do 1 lane first, and then we can quickly tell them whether or not it's worth the time to sequence the remaining 19 lanes, or if they should hold off and wait for things to wait for an investigation before we proceed, or maybe an optimization, or maybe a step needs to be done. It's universal, so regardless of the protocol method, it still gives us a number, so it doesn't need to wait for that. It can detect things like reagents and spike-ins, so it doesn't necessarily need that reference genome to be available. And then we can also do a stage where we scan and optionally remove microbial reads from the sequence if the library is going to do something simply, things like that. And it's consistent. So metrics are generated and loaded automatically into a database. And so we do this on every single lane, so it's always stored, so we can look at it historically. And that really forms the basis for our sort of future review, our retrospective or historical comparison, and our trend analysis. So the QC0 pipeline that we use, it basically has every single lane gets analyzed for a standard set of metrics. So some of those metrics are going to be used for the QC pass or fail assessment, but then all of the metrics that we collect are stored in a database which we can do for future comparison or historical review. So the sequence comes off the instrument itself, usually in the form of FASTQ files, and then we process these on the BioQC cluster and store them in a Postgres database. So our database contains, and when I checked this morning, I think it had 200,000 libraries worth of data in there. And so that's really what we can use to do our longitudinal excuse me, trend analysis. But also, obviously, our database proceeds into our automated assessment pipeline, where we can then make a decision whether the lane is approved or rejected. And so the automated assessment pipeline does that. Now, occasionally the pipeline encounters things that it can't deal with. I mean, science is always changing, and so we always get new things and new looks, new types of data and new protocols coming through. So that stage then goes through a manual review. So a person will actually look at our metrics and decide whether or not, and then make a human judgment on whether or not that's approved or rejected. Okay, and I'm just making sure everybody can still see my slides, I hope. So what are the type of things that you can look for without actually aligning it to the data? So some things you can look for. You can look for reagent content, so you can detect sequences that might contain adapters or vectors, some of the standards that are put in, or ladders.
Hi, Kane.
Yeah.
If I could just interrupt you for a second. It's Garth Evans here, support tech. We got a participant, Gustavo, that accidentally shared his screen. You're back, Kane. I hope everybody can see your presentation. You were just off momentarily, but sorry to interrupt you. I'll get you to pick up where you left off.
Sure. Thanks, Garth. So just to start again here. So what can we look at? What type of things can we look at without actually aligning things to the reference genome? So we can look at reagent content, detecting whether or not a sequence contains adapters, vectors, standards, or ladders. We can look at microbial contamination. So we can use a read classifier, something like a BioBloom tool, to detect specific microbial contaminants. So in our pipeline, we actually look for 45 different species or different microbes. We can look at index and pooling problems. So we can examine the no-match bin, the index no-match bin, and see if it contains an unusually large number of reads. or we can look for expected indices that are missing reads. So if you put in five indices into your, or 5 libraries with 5 different indices into your library, but you only see reads from 4 of them, we can identify the one that's missing. And of course, without alignment, we can also do things that look at sample swaps. So we can compare variant calls between samples of the same individual. We can look for spike-ins that have been put into the sample, and I'll talk a little bit more about this later. And we can also look at the distribution of indices to match what was pooled, which is somewhat similar to the previous point, but I'll go over an example a little later on to demonstrate why it can also help us to determine a sample swap. So for sample swaps, as I mentioned, we sort of have 3 methods of detection. So we have the SNP concordance, which is for human libraries only. We have a set of what we call spike-ins that we've generated in-house, and we use these on the following 4 protocols: the whole genome, RNA-seq, amplicon, and whole genome bisulfite. And then we can also look at the index splitting reports. So for SNP concordance, we actually have a bioinformatic implementation of AFI's 500K chip array. And basically, it looks at the individual read sequences. It does not align them to the genome, but actually looks at downstream sequences of the variant position. And then we can actually produce a concordance report between different samples, between different runs, or between different sublibraries. So I've kind of pasted an example of a SNP comparison table here. And you can actually see very clearly that when you do a pairwise comparison between all of the runs of from this particular individual, you'll see there are 5 runs here. You can actually see the red there clearly indicates which sample is the outlier. So that's the sample of the library that doesn't match the other lanes of its that were supposed to have come from the same individual. So that allows us to quickly pick out which one was the outlier, and therefore which one is probably the one where the sample swap took place. Another protocol that we use is the method that we use is the spike-ins. And so these are 200 base pair oligos that we've created, and we actually aliquot them into every sample at tiny amounts when it arrives at the GSC. We try to aim it so that it does approximately 10,000 reads, and so in the actual final sequence data. So typically in an Illumina run, you'll get probably millions of reads to hundreds of millions of reads, how you've the depth to which you've sequenced. So 10,000 reads is really a fairly small amount, and then we can use those to detect how much of our, we can look for those oligos and determine the purity of our oligos to see has there been any sample swap taking place, or we can use that to determine if there's been any contamination by looking at what oligos we didn't expect to see in there. So a lot of our quality control metrics can be categorized into a number of different categories about what they tell us. So as you can see in the top left there, some things will tell us like adapters, reagents, or dimers, or the duplicate rate or contamination, or the PF rate will tell us about the sequencing quality of our library. Others, things like bisulfite conversion rate or chip capture, will tell us the success of laboratory processes. We can determine the sample degradation by looking at RNA degradation, or we can look at the fragment size. That might give us information about our sample itself. We can also look at the historical comparisons. If we sequenced this previously, sorry, I'm getting a little bit of feedback here. I'm hoping somebody maybe doesn't have their microphone muted, if that's possible. Okay. Well, I'll try to keep going. Anyway, so through historical comparison, if we've sequenced some lanes from previously, maybe this is a project where a certain sample has requested that 3 or 4 sequence, we can compare that historically to make sure that the lanes are performing, giving roughly the same style of metrics. Sample identity, of course, I talked about that in the previous slide. And of course, we also have a number of metrics that also tell us about gene complexity and library diversity. So we can look at how many microRNA species we observe, the number of genes we detect, intergenic content, or intron-exon ratio. Of course, this list is not comprehensive. We really follow way too many metrics for me to list all in a single slide. As I mentioned, we have upwards of 60 at the QC0 level alone. So, but this is just an idea that different metrics can tell you different things about your sample. So how do we decide when something passes or fails? Basically, this is determined on the in the lab. What we can do is we make that assessment based on what happens on the instrument itself. So the lab staff kind of go through the machines and they look at the reports that come out of the Illumina software, and then they sort of use their own personal experience to determine whether or not something can pass or fail. But on the bioinformatics side, with our automated pipeline, with everything going into a database, what we can actually do is we can take all of our statistics and then group them and make histograms, and then use the 95th or 99th percentile to set warning and failure thresholds. So in the graph I've shown here, you can see that most of the population appears in the white area, and the blue vertical line is actually this specific library that we're looking at, which is well within where the population lies. But out here in the red area, you can see those would be libraries that we would fail because they look outside of what 99% of our libraries look like. And the yellow area, and hopefully that's coming through, but that's right about here, just a little small section just before the red area near the middle of the graph, is where we would see warnings, where things are outside of the 95th percentile of everything. So it's important to distinguish between metrics and thresholds. So metrics is a measured or calculated characteristic of a library, whereas a threshold is a value at which a library is to be assessed for quality. So the key really here to remember is that not all metrics have thresholds. So some examples are read count, for example, or whether or not our expected spiking is observed. And then there are other metrics that do have thresholds. So things like reagent leftover, contamination rate, or alignment rate. And also the thing another thing for us to understand is, of course, between hard thresholds and outliers. So hard thresholds for us are the absolute point at which a library must be failed. And usually this is an indication that something has gone severely wrong, maybe whether it's on the bioinformatic end or the lab end. So some examples might be something like something within a very, very low alignment rate, so let's say less than 60%, or something with a very high contamination, something with greater than 50%. So these are things where something has gone wrong that we really need to investigate. That is that really what is a true representation of the sample before we send it out, or is it something that we need to take a look at? It's something that we might be able to correct on our end. The other thing to look at, the other way to look at it is outliers, and these are things where sort of in that yellow warning area where the metric is beyond the 95th percentile of our historical GSC data. And typically these might be cases that contain usable data, but it's less than ideal. So some examples might be low quality or low input material, or slightly lower genomic coverage. So these are things that, you know, it might have arrived at the GSC that way in terms of like it was just a sample, it was the best that the investigator was able to provide us. So even though it doesn't give great data, they still want to look at it. And so typically we will always review any library that has more than 3 outlier metrics just to make sure that everything's on the up and up and, you know, it is looking the way that it should and that this is still valid data. So why is quality control important? So here's how what I call the organizational benefits, or how it helps you internally within your group. So there's the obvious one, which is to confirm sample identity. We can use our quality control metrics to determine if a sample swap or a contamination event has taken place. It helps us to detect problems that may have occurred in our laboratory process. So if there was a case of uneven pooling, or where something, an RNA library has unusually high ribosomal content. It helps us to make improvements to protocols. So because everything runs through our system, all of our lanes, and they always get put into our database, we have a historical reference so that when we do something new, we can actually compare the new version to the old version. It helps us to compare our results to previous experiments, so we can look at batch effects that may slowly creep into the data over time, or whether we need additional lanes or how many lanes we might need. And it also helps us to reduce costs by avoiding analyzing bad data and integrating it into our results before an existing result, so we don't have to separate it back out later. How is it beneficial to your science? So as a QC gate, Obviously, again, it prevents bad data from being incorporated into your analysis and being potentially uploaded into a repository in an archive. With sample swaps and low library diversity, it helps you to identify outliers. So if we let you know, or if your QC reveals that something's out of range, that might help you when you're writing up your results and sort of discussing, oh, well, we noticed this, and this is the reason why. So that it helps you when you're publishing to explain what you're seeing. And finally, it can also help you to perform a trend analysis over time. Again, looking at your results and then identifying areas where you could make your protocols better in terms of optimizing your lab process or improving your bioinformatics pipelines. So now I'm going to look over some examples of things that we've observed different case studies and anecdotal examples of QC. So as I mentioned earlier, here's an example of index splitting. And so in this particular case, we've sequenced this library that has 4 expected indices. And when we look at the breakdown of where the reads come out, we can see that the first 3 have a good number of indices, but the 4th one, indicated in red there, has 0% of the reads. So, and at the same time, the no match bin, so where it doesn't match the expected indices, has sort of the remaining group. So what we can conclude from this is that 4th sample with index TGA CCA obviously has not been pooled into the library. And we can further rescue that data by going into the no match bin and looking at what might that missing index have been, and then basically resplit the library on that basis. Now here's an example of index splitting where we've sequenced 3 lanes. They're all library A, so it's the same sample that's been sequenced in 3 lanes. And what we can see is that while the first lane and the 3rd lane look good, the middle lane there has a distribution that looks a little bit off, quite unusual. And so what we can conclude from this is that lane 2, being the outlier, has been swapped with another lane. So here's an example of a sample swap that we can identify using the index splitting report. Now sometimes we actually have libraries that fail QC, but the data itself is still usable. And I've listed 4 cases here that we see where there are samples that, they do fail our QC metrics. They don't look like 99 or 95% of the libraries, but actually the library is still good. The data itself is still usable or valuable to the study itself. One example is with FFPE samples that we see. Sometimes there's degraded DNA that comes through, which means that PCR amplification was needed to get enough material to put it on the sequencing instrument itself. But typically what we'll see is significantly higher duplicate read rates because of these amplifications that are required. In a second example, for amplicon libraries, we see that if the amplicon sizes are small, so our Illumina machines, the most common read length we use is either 150 or 250 base pairs. If the amplicons are smaller than that, then the instrument itself will read through the insert and go through the other end where it'll start reading the Illumina adapter. So again, our QC report might tell us that we're seeing a lot of reagent in it. But again, because we know that our amplicons are smaller and we expect read-through, the data is still good from the amplicon itself. So we wouldn't fail the data. We'd still indicate that it's failed our QC metric, but the data is still valuable to the downstream, to the study. Another example are metagenomic studies or xenograft libraries where we'll see a lot of off-target species reads. Again, that doesn't necessarily mean it's bad because it's in a xenograft, so you know you're going to have or in a metagenomic study where we're looking for reads from a large number of species. So obviously that is, again, valuable to the study itself. And finally, with low input libraries, we just naturally have a lot of background that gets amplified. And so that might be lower fragment diversity. But once again, if it is low input, we knew that coming in, means that the data is still good and usable. And of course, we have our last category, which are our safety checks. And so that's with when we should fail QC, and this is a reason to stop because we think that something might have gone wrong that could be corrected. One is a low alignment rate. So one anecdotal example we came across was with, of course, BWA, the ALN version of BWA doesn't work very well on reads that are longer than about 125 base pairs. And so if that's been used accidentally, then obviously we should switch to using the more appropriate BWA-MEM version. Another case would be if we had aligned it to the wrong reference genome. Maybe it was incorrectly specified to us. Sometimes we have, obviously, for sample swaps, you don't want to publish or analyze data. So that's another example of where you want to stop, where something fails QC and you want to stop and not. Low bisulfite conversion rate of the lambda phage. So the lambda phage is something that's used in bisulfite conversion reaction to measure the conversion, whether or not the reaction went to completion. So that would be an example if the conversion rate is low that we want to stop and sort of maybe repeat that section of the experiment and see what happened. For RNA-seq libraries, if you have too much genomic DNA in your library, that might affect your calculated expression level. So again, that's a reason to just kind of take a stop and look at the data and see whether or not it's affecting your results. So that really brings me somewhat to the end of my presentation. So basically what we talked about today is what is quality control. I talked about the 3 levels of QC that we look at and how we do it at the GSC. So in terms of we have things at the laboratory stage, we have an automated bioinformatic pipeline that helps us to review things quickly and more systematically. And of course that deposits QC data from every lane that we sequence into a database that can be used to review for historical purposes and as well as to examine trends. And that some data that fails QC can also sometimes be used depending on the nature of the investigation and the scope of the experiment. And finally, I try to hopefully impart to everyone how QC is useful, not only for the work that you do in your organization, but also for the science that you do. How it saves time in your data analysis, how it aids in the interpretation of the results or the data that you see in your publication, and how it identifies trends and areas that you can improve or improve whether it improves your protocols or improve your bioinformatic pipelines. So just to go through my acknowledgements, and I don't know if anybody has any questions that they'd like to ask. Now would be a good time. I'm near the end of my presentation. Thank you.